States, UK, and France and Germany, who were the main contributors in the research of microbiology, all were involved in war. Okay, but the discovery of penicillin motivated all the researchers to find uh, newer antibiotics, okay, for the infection. And as you know, there was a heavy, we can say, commercial benefit also behind it. So all the pharmaceutical research companies and all the, we can say, uh, academicians also, they were all interested in finding out newer antibiotics or newer sources of antibiotics. Okay, at that time they had no idea of chemical modification. Okay, although the structural elucidation of penicillin and structural elucidation of other chemicals was also underway. Okay, so there was a researcher by the name of Paul Ehrlich. Paul Ehrlich found an inorganic compound made up of arsenic. Okay, for the treatment of syphilis. Okay, he discovered a chemical compound. So we can say the chemotherapy also started. Chemotherapy, the basis of chemotherapy was laid by Paul Ehrlich in 1910. Okay, now what was why we called from 1943 till 1970? Why do we call this era of microbiology the second golden age? Okay, it started after the Second World War. Okay, 1943, the Second World War ended, and there are some important inventions and discoveries which we can say really revolutionize the science of microbiology and laid the foundation of molecular biology and genetic engineering. Okay, so what were those important inventions? First was the invention of electron microscope. Okay, electron microscope actually revealed the structure of cell in detail, and we were first able to find out that the nucleic material, genetic material in prokaryotes is no more enclosed in the nuclear membrane, and in eukaryote it is enclosed in well-defined nuclear membrane. So the two groups, prokaryotes and eukaryotes, were developed. Okay, and the difference was a difference in detail in cellular structure, in the structure of organelles, was revealed by electron microscope. Okay, at the same time in 1943, two microbiologists, okay, from Italy, and by the name of Salvatore Luria and Max Delberg, both of them worked on bacteria as experimental model, as we work on mice or rats or dogs, or for some microorganism for some infection in case of chimpanzee also. But what they did, they used Escherichia coli, E. coli, a gram-negative bacteria, as experimental model. Okay, and they were the one first to observe mutations in genetic material, and they noted that mutations led to the resistance to viral infection. Okay, when they used bacteria as animal model, other biologists also used the same model. Okay, and literally I can say that bacteria has served a lot mankind to let them understand the human, uh, the researchers found all the, we can say, basis of protein synthesis, enzyme function, function of mRNA, the function of tRNA, the protein synthesis, how it is carried out on ribosome, all they learned from bacteria, and their understanding of these mechanisms led to genetic engineering. Because in 1953, almost 1953, it was established that DNA is the most important control center of the cell, and DNA is the most important genetic material which decides about the fate of it. It was not by bacteria. Two researchers, Beadle and Tatum, were working on a fungus. Okay, they were working on a fungus, and I told you, in the meanwhile, search for antibiotics is also, for antibiotics we were also working on microorganisms. So microorganism, we can say, were the center of research. Okay, microorganism for understanding of genetic structure. They found that one gene is responsible for one enzyme, and enzymes are responsible for protein synthesis, and microorganisms are independently synthesizing their own proteins. They do not take up everything like us or like animals. They do not take prepared protein, okay, in their food. They actually synthesize their protein themselves. They synthesize their nucleotides themselves. Okay, so they are quite independent, but because they are independent, they have a lot of enzyme, important enzyme for that purpose. Okay, so the discovery of DNA, the discovery, the understanding of in later on the structure of DNA in 1960s was revealed, and how enzymes, how mRNA, tRNA are responsible, rRNA are responsible for protein synthesis, how proteins are synthesized, and how it is controlled by a particular gene. Okay, after the structure of DNA was revealed in 1960s, it was established that DNA is controlling the synthesis. One gene is responsible for the synthesis, and we can take out that gene and we can place that gene. It can be taken up by a bacteria, and if that bacteria is provided with all the nutrition required, it will synthesize the same protein. Okay, during that same era, that is late 1920s and early 1930s, insulin was also discovered as the lack of insulin was discovered as the cause of diabetes. Okay, in the same era, and all the experiments were carried out on dogs. Okay, they were the pancreas was removed from the dog's body, and they get all the symptoms of diabetes. Okay, a whole syndrome was developed, because diabetes is not a disease. We can say, it is called it better should better be call it syndrome. Okay, because it affects whole body. So it was found that insulin is responsible for the lack of insulin, or malfunctioning of insulin, or malfunctioning of pancreas is responsible for diabetes. Okay, so this era is marked with the invention of electron microscope, and how electron microscope change our understanding of microorganism, and the understanding of protein synthesis, and the structure of organelles, and the structure of DNA and RNA. And later on, we were able to understand the nucleic acid sequences, that is the structure of DNA, RNA, and protein, that is amino acid sequences. We found that enzymes are actually protein, and we found that enzymes are actually synthesized by microorganisms themselves, and the method was understood by using the microorganisms as a model, and that laid the foundation of genetic engineering. So genetic engineering, okay, and the foundation laid to for the development of science of molecular biology, because we were able to understand the biochemical reactions at molecular level. Okay, with the help of electron microscope, and because of the better understanding of biochemistry of microorganism, which led us to work on higher living organisms also. It helped us in understanding a lot the biochemical reactions going on in microorganism and then on in living cell of mammals or higher living organism. Okay, so later on, in 1940, I told you penicillin was made available, and all the pharmaceutical industries were following uh, uh, they were following uh, we can say search for newer antibiotics, and it was really exciting. In 1944, a scientist Waxman discovered uh, streptomycin, and streptomycin is an antibiotic which was first used to treat the uh, TB patient. Okay, it was the first drug which cured TB. So it was again it was an amazing discovery, and I told you, because all the researchers, all the pharmaceutical company found that there is a commercial benefit also behind the research of antibiotics. So they gathered from almost from the whole world, they gathered 20,000 samples of soil. Okay. They were looking for fungi and they were looking for actinomycetes. Actinomycetes, remember actinomycetes were early were considered as a group of fungi, but later on, when the classification was based on 16S rRNA, actinomycete group, which is the largest antibiotic producing group. Okay, streptomyces species. The genus is Streptomyces, and many species are producing, for example, erythromycin, chloramphenicol, tetracycline, and many antibiotics, except gentamicin. Okay, neomycin, kanamycin, all are produced by actinomycetes. They were looking for actinomycetes in different samples of soil from around the world, and a number of in late 1940s and earlier 1950s, a number of antibiotics were discovered and worked on to find out their benefits in different infections and whether they are safe in human being or not. Okay, so most what you will describe, or what you will tell me if I asked you, just tell me about revolutionizing discoveries or inventions of second golden age of microbiology, and which one had what type of we can say impact, okay, or profound impact on the development of microbiology. We must mention the discovery of penicillin, or the availability of penicillin in the form of injection, and the invention of electron microscope, and the development of genetic engineering after the structure of DNA was elucidated, and the structure of mRNA, tRNA was elucidated, and the understanding of biochemistry or biochemical reactions going on as the basis of life in living organism. So this second era, golden era of we can say microbiology had many important, I must say, aspects, okay, in development, and all findings, all discoveries were revolutionizing, all were very important in the sense that they had a profound impact on the health, or we can say on the treatment or an understanding of uh, the biochemistry of living organism, how proteins are made, how energy is produced, how mitochondria is used as a powerhouse of living organism in eukaryotic cells, and how microorganisms are beneficial for us, how we can use them not only for producing antibiotics but for producing, because after this genetic engineering was developed, then only we were able to find out the DNA recombinant. We started working on DNA recombinant technology. Okay, the products of DNA recombinant technology were not available till 1970s. Okay, the first vaccine came into the market in 1980s, that is hepatitis B vaccine was made available. And for insulin, it was important to work out on DNA recombinant insulin, because before that, insulin was extracted from animals' pancreas. Humans, you cannot made a human, you cannot use the pancreas of dead humans. You have to get pancreas from live animals. So two types of insulin, animal from animal source, that is from pork and from beef were available. Beef insulin and porcine insulin. So the porcine insulin was not accepted in, we can say, 40% of the population, that is Muslim countries. Okay, so it was a challenge to market uh, porcine insulin. So they worked on DNA recombinant insulins, which is now being made by E. coli, the gene from the human pancreas, from human beta cells, cells of islets of Langerhans. Okay, the gene was isolated and it was transferred into the E. coli. Okay, and E. coli took up their gene and started producing insulin, which is then purified, extracted and purified and made available in form of injection, and which is acceptable throughout the world. Okay, nobody is rejecting, because it is the source. Nobody is saying that the source is from any forbidden animal. Okay, so we can say after 1970, the basis of biotechnology, that is genetically engineered microorganisms, which were producing not only insulin but growth hormone, and we also extracted many acids like citric acid is being made by bacteria for us. There are many enzymes like amylase, lipase, okay, invertase, lactase. These are all made by microorganism, and we are using these enzymes in, for example, food industry, in confectionaries, and for example, lipase is protease is used in, for example, laundry, okay, in washing of clothes. It is being added. If you have seen in the advertisement, also the enzyme action. Okay, these enzymes are produced right now by bacteria for us. We are not only, still we are getting penicillin from the Penicillium chrysogenum. Okay, we are still extracting kanamycin, that is the source of we can say aminoglycoside, that amikacin. Okay, amikacin is a semi-synthetic aminoglycoside. So we are still getting antibiotics from microorganism. We are getting from genetically engineered microorganisms. Also, we are getting human products. Okay, and these are much more, we can say, pure. They are as human as they should be. Okay, as we require, and there is no controversy about forbidden animals. Okay, now we still have many challenges, because the role of microorganism in environment has also been understood. Okay, we got a newer classification which was based in 1970. It was based on 16 srRNA, because we have elucidated the structure of RNA and DNA. So the scientists thought that instead of classifying the microorganism on the basis of some, because microorganisms do not have as many, we can say, morphological structure as higher animals or plants. Okay, they do not have roots, they do not have leaves, they do not have flowers. They only have their cellular structure and their appearance. So under microscope only we can observe these. So they should be classified on the basis of something very, very, we can say, concrete, which is not changing very frequently. And as we know that bacteria undergo mutation, so it was difficult to find, to make the classification on the basis of DNA. So scientists find out that 16 srRNA sequence, okay, as it is a nucleic acid, so the sequence is based on nucleotides. Okay, so it's because it is such an important part of microorganism, that and other higher uh, organism also, that its sequence cannot be changed. It cannot afford, a microorganism cannot afford a mutation in it. Okay, in this ribosome 16s rRNA, ribosomal RNA structure. So that's why micro, because it does not undergo mutation, and because it is responsible for very important, we can say, housekeeping enzymes, which cannot afford mutation. Okay, so scientists selected this 16s ribosomal RNA as the basis of classification, and that also happened in 1970. Okay, so right now we have some very interesting new finding also. I told you that DNA recombinant vaccine was developed for hepatitis B, and it was made available in 1980s. But with the advent of microbiology, newer challenges were also on the edge. For example, the resistance. ओके बेटा 9:20 अभी हुए नहीं है मेरे पास. Okay, okay, we will inshallah we will discuss in our all the we can say important points of second golden age I have discussed today, and for third I will give you some points in our physical class, inshallah. Okay, thank you so much, and I have taken your attendance